So, ladies and gentlemen, welcome. This is a kind of joint presentation for advanced biochemistry and enzymology on enzymes, coenzymes, cofactors, and prosthetic groups. So, I would start that there are some enzymes which can simply work just with a protein structure, with protein chemistry. So, the active site is composed only of the protein chain, of the amino acid side chains and the main chain, and this is only this is enough for the catalysis. There is no need for some special chemistries, and there are many enzymes like that. But there are also enzymes which require some special chemistry, and they need either some covalent modification of the active site by some non-protein groups, or they require some non-covalent binding of some compound which also somehow participates in the catalysis. And I would call these compounds like prosthetic groups. And besides this, we also have a group of small molecule compounds which play important role in enzyme function, and these are coenzymes. Coenzymes are like regular substrates for enzymatic reactions, but in a cell they are in a relatively small quantity and they are being like continuously recycled. So there are enzymes which convert coenzyme from one form to another. Sorry, this is the cat is trying to catch my headphones. Sorry. So coenzymes are permanently being like recycled. So there is one reaction which converts a coenzyme from form A to the form B, and another reaction which converts B back to A. And both these groups and also some minor exceptions are like grouped to the term cofactors, and the important term which is also linked to coenzyme, cofactors and prosthetic groups are vitamins, because many vitamins are like either used as coenzymes or prosthetic groups, or they are somehow modified to become coenzymes or prosthetic groups. So here is a kind of definition which I somehow explain, and here is the list of vitamins for humans. And I would like to mention the fact that this terminology is a little bit fuzzy, because like the way how different coenzymes, cofactors and prosthetic groups were mostly discovered is that somebody took some extract from cells and analyzed it by some analytical chemistry methods some many decades ago, and it was found that it contains some small molecule compounds, and later it was found that there are some enzymes which, if you isolate them, they do not work very well, but if you add such a compounds, they start to work. So that's how these molecules were discovered, and I would say much later it was found that some of them work as prosthetic groups, some of them works as coenzymes. So this somehow brings some fog into the terminology. And vitamins were simply discovered as compounds which are present in food, and if you have a lack of vitamins, this causes some health problems. And they have a small daily intake, so they are not like directly involved in some energy metabolism, for example, like some essential amino acids or essential fatty acids. They have to be consumed in a relatively high amounts compared to vitamins, and they are somehow like undergoing some changes, chemical changes, but vitamins can be somehow converted to coenzymes, cofactors. Not all vitamins, but majority of vitamins are either used in the cell as cofactors or some precursors for cofactors. And I have to mention that like this terminology I use right now is not somehow 100% general, but you may be used to some different terminology, so I will not force anybody to this terminology, but I find it quite logical, this separation or this classification of cofactors as coenzymes and prosthetic groups. And here is, I should maybe explain more in detail these prosthetic groups and coenzymes. So like imagine that you have, I will give an example. So typical coenzyme is NAD+, which is a coenzyme of oxidoreductases. So it's like natural oxidating agent which can oxidize some substrate, and it is converted from the oxidized form NAD+ to the reduced form NADH. And if you imagine like that there is in the body only a reaction in which NAD+ is being converted to NADH, in a very short moment such a cell would run out of all NAD+ and all the NAD would be in NADH form, and the metabolism would stop, or this part of metabolism dependent on NAD+ would stop. So there must be another reaction or another reactions which somehow convert NADH back to NAD+. So there is one enzyme which converts NAD+ to NADH, and then other enzymes which convert NADH back to NAD+. So to give some examples, so you probably know from glycolysis there are some enzymes which convert NAD+ to NADH. So to give example, like glyceraldehyde 3-phosphate dehydrogenase. So this the reaction catalyzed by this enzyme is like oxidation of glyceraldehyde 3-phosphate to bisphosphoglycerate with the input of phosphate anion, and NAD+ is converted to NADH, so it's oxidative reaction, and NAD+ is reduced to NADH. And then in glycolysis there is no reaction which converts NADH back to NAD+, but if there is glycolysis going under anaerobic condition, for example in ethanol fermentation or during some physical activity with a lack of oxygen, this NADH is converted back to NAD+. So in case of ethanol fermentation, it is the reaction of acetaldehyde being reduced to ethanol. In case of the physical activity with lack of oxygen, this would be conversion of pyruvate to lactate. So there is one reaction, one enzyme which converts coenzyme from the state A to state B, and there is another reaction or other reactions which do the opposite. And of course, like there are some, not so many, but there are some coenzymes which can exist in more than 2 different forms, like folic acid. Folic acid is the example I will show a little bit later. And what about prosthetic groups? So prosthetic groups, they are again like converted from one form to another and then back to the original form, but this all happens in one enzyme. There are some like prosthetic groups which can somehow dissociate, but important is that they do not have to dissociate, so they can be like if they undergo the one catalytic cycle in the enzyme, they are converted from like original form, from the form A to the form B, and in the same catalytic cycle from B back to A. So to give an example of a prosthetic group, so the common prosthetic group is FAD, flavin adenine dinucleotide, and FAD is converted, for example, one enzyme which uses FAD is succinate dehydrogenase in Krebs cycle. So succinate dehydrogenase, the first kind of sub-reaction is that succinate is oxidized to fumarate and hydrogens go to FAD to give the product is FAD FADH2, the reduced form, but without any dissociation in the same catalytic cycle in the same enzyme. This FADH2 is converted back to FAD, and there are protons released and electrons go to the second like sub-reaction in which coenzyme Q is reduced. I hope this is quite clear, like the difference between coenzymes and prosthetic groups, and altogether I would call them cofactors. I must stress that there are different people using slightly different terminology, so sometimes you can experience the terminology in that the people use the term prosthetic groups for something which is covalently bound to the enzyme, and cofactors could be something which is not covalently bound to the enzyme, and it's something for which I would also use the term prosthetic group. So I don't force you my terminology. Feel free to use the terminology you are used to, but I think it's very important to somehow remember this difference, that these compounds are being like recycled by 2 different reactions, whereas some are like integral parts of enzymes and are being recycled within the catalytic cycle. I also wanted to mention, no, that's all. So I will start with the first one, with NAD. So here is the structure. This is the structure in an oxidized state. Here everything important happens here. So this is like in oxidized state. Here of course there is a hydrogen here, so I drawn it here like explicitly, and when it's being reduced to NADH, this reaction happens. So the hydrogen in form of hydride particle like comes here and binds here, and because it was hydride like H minus, then the total charge at the end is 0, because here there was a plus charge, you see that there are four bonds on nitrogen, so it has plus charge. And here with this hydride added, there is like a double bond, two double bonds here. It's no more like a aromatic ring. There are two hydrogens. Of course, one is like they are not in a plane, but like if these carbon atoms are in plane, one is like towards us and one is in the opposite direction. And there is no plus charge anymore because there is like 3 bonds on nitrogen. There is a kind of very similar coenzyme called NADP+, and the difference is that here this R is not hydrogen but phosphate group. There are some enzymes which are strictly there are some oxidoreductases which strictly use NAD+, there are some which strictly use NADP+, and there are some which can use both of them. Here in this slide, it's quite interesting that some people, by using, I think, NMR, if you use a substrate which contains deuterium instead of hydrogen, and then you analyze the produced NADH, or maybe we should call it NADD because there is deuterium instead of hydrogen. So if you study this NADH, or let's call it NADD, by NMR, you can somehow find from which direction, whether like from like our direction or from the opposite direction, this deuterium or this hydride H- or D- anion like came. So this can be used to somehow study the mechanism of the enzyme. And very important fact about NAD+ or NADP+ and NADH or NADPH, the phosphate here really doesn't matter, is that the reduced form absorbs at the wavelength of 340 nanometers. So in UV range. So this is like the spectrum, typical spectrum for NAD+ or NADP+ in a like gray, and NADH or NADPH in black. And there is a big difference in absorbance at 340 nanometers. And this is a very important fact because it is very popular in enzymology, in enzyme assays. It's also very popular in clinical biochemistry, in assays of different analytes in blood or different enzymes in blood, because you can, using spectrophotometry, you can distinguish between the oxidized and reduced form, and you can measure the rate of reaction simply by spectrophotometry. So you insert the cuvette into the spectrophotometry machine, and you can continuously measure the increase of absorbance or decrease of absorbance at 340 nanometers. It's possible to use something which is called coupled reaction. If you have some reaction where NADH or NADPH is not like neither produced nor used as a substrate, so it's possible to use like some series of reaction which end up in some production, for example, of NADH. And I will show these tricks next week, because they are quite useful and quite commonly used in enzymology. Here I have one comment that the NAD+ plus can be a prosthetic group of S-adenosyl homocysteine hydrolase. So S-adenosyl homocysteine hydrolase is the enzyme which hydrolyzes the bond between sulfur and carbon. So it's like some thioether bond. So you can imagine from organic chemistry that it's not a very not simple task, like to hydrolyze some sulfur carbon bond. And the trick which is used by this enzyme is that it first oxidizes this substrate, this S-adenosyl homocysteine. This substrate becomes like easier to be hydrolyzed, so then it's hydrolyzed, and then in the same catalytic cycle this product is again like reduced. And this NAD plus starts in the reaction as NAD plus. It is converted to NADH in this first half of the reaction, and without being dissociated, it is like converted back from NADH to NAD plus in the second part of the reaction. So it's typical prosthetic group, but in 99.9 like situations, NAD plus or NADP plus is a coenzyme. So the second one I have already mentioned is FAD, flavin adenine dinucleotide. Maybe I forgot to give like the full name of NAD plus, so nicotinamide adenine dinucleotide, and NADP plus is the same but with phosphate. So here this is flavin adenine dinucleotide. The name is a little bit confusing because here if we have NAD we got like 2 ribose sugars, but here instead of 1 ribose we have it's a reduced form called ribitol. And there are different possible chemistries going on FAD, or flavin mononucleotide is like kind of truncated version of FAD and is also used by some enzymes. And the most common like chemical reaction which happens is that these hydrogens are, one is placed here and one is placed here, and there is some rearrangement of double bonds. So instead of 2 double bonds there is 1 double bond. And there are some other possibilities how FAD can participate in the reaction. There is also a possibility to like distinguish spectrophotometrically between the oxidized and reduced form. It's possible to use spectrophotometry to measure whether to distinguish between FAD or FADH2. But this is not very useful for measurement of enzyme or for enzyme assays, because FAD is typical prosthetic group. So it stays bound on the enzyme, and it's not like a regular substrate like NAD plus is. So if you have some reaction which is catalyzed or which in which FAD participates, for example, I mentioned succinate dehydrogenase. So in one catalytic cycle, this FAD goes to FADH2, but in the same catalytic cycle, it goes back to FAD. So it's like concentration of FADH versus FAD versus FADH2 does not change through the course of reaction, and maybe you can use it to study the enzyme mechanism. So for example, you can use this to study whether the enzyme is like running 100% and it's mostly in this reduced form, or maybe it's like resting a little bit and it's in the oxidized form. Something like that is possible to study in mechanism of enzymes which depend on FAD. but it's not possible to use it in enzyme assays because it's prosthetic group. There are some other interesting cofactors, those which are like I did my best when I prepared these slides to somehow follow enzyme nomenclature. So I started with oxidoreductases, and I wanted to go through transferases and to cover all these classes of enzymes. So it works at the beginning, but later it becomes some kind of like there starts some mess, because some prosthetic groups and coenzymes are somehow like scattered across different classes of enzymes. But here we are still in like we are still in oxidoreductases, so quite interesting cofactor is quite interesting prosthetic group is lipoic acid. This one is also like prosthetic group from this alternative terminology, which uses the term prosthetic group for something which is covalently bound, because here there is like this represents the enzyme. So this carbonyl group, carboxyl group is attached via amide bond to a side chain of some special lysine in the structure. And you may know from biochemistry lipoic acid from pyruvate dehydrogenase complex, where like if the substrate is pyruvate, it's first decarboxylated, and this decarboxylation product, which is acetaldehyde, is first like bound to lipoic acid. So here, if it's acetaldehyde, there this would be like methyl group, and then this acetyl group is transferred to coenzyme A. I will introduce coenzyme A in a moment, and this lipoic acid is reduced, and then it can be oxidized back by like joining these sulfur atoms. Quite interesting group of coenzymes of oxidoreductases are chinones. So the famous one is ubiquinone, coenzyme Q, and some plastochinones are like some plant counterparts of ubiquinone. So this can exist in oxidized form, which is this one, or a reduced form, which is like the hydrochinone. This is just like what happens on this aromatic core. And it's quite interesting that these compounds are like similar to, for example, NAD+. So again, it can exist in oxidized and reduced form. It's a coenzyme. It is oxidized by or reduced by one substrate, by one enzyme, and oxidized back by another enzyme. But what is interesting and what is different for these chinones is that these molecules are highly non-polar, highly hydrophobic. So they have very long chains here. So they are completely hydrophobic. So instead of being like dissolved in the space of the cell or in the cytoplasm or in the mitochondria matrix, they are somehow like dissolved in this two-dimensional space of the membrane. So quite interesting cofactors, which could be coenzymes as well as prosthetic groups, are pterines. The most famous one, tetrahydrobiopterine, participates in a reaction of oxidation of phenylalanine to tyrosine, in which like molecule oxygen is used, so is dioxygenase. and monooxygenase, and one of its oxygen is then introduced to tyrosine as this OH group, and tetrahydrobiopterin is oxidized to dihydrobiopterin. So from this form to this form, there is some again some rearrangement of double bonds. This reaction is quite famous because there are some people with like a reduced or lack of activity of this enzyme due to inborn error or inborn mutation, and this is the most common like inborn error in metabolism called phenylketonuria, because patients with which cannot like do this reaction, their enzymes are broken by mutation. These patients like excrete or some alternative product of like phenylalanine, and this causes some quite severe syndromes. So usually these people have to have a special diet in which they consume like all so they consume like all the food with a low protein content, like a low protein diet, and this is supplemented by a mixture of separated amino acids of all 20 amino acids except phenylalanine. So they have to somehow avoid consumption of phenylalanine as much as possible, and it's also written in on every like can of Diet Coke, which is sweetened usually by aspartame, artificial sweetener, which is a peptide which contains phenylalanine, and this is so there is a warning that people with phenylketonuria should not drink it. And there is also quite interesting like pterine, which is also in human body and which contains a molybdenum atom in its structure. So we have molybdenum in our body and in our metabolism and in our enzymes. Heme is quite interesting molecule. I was talking about heme in advanced biochemistry when I was talking about oxygen distribution in body, but it's not the only role of a heme. It's present in many different enzymes where it participates in catalysis. I would say like metals are very popular in catalysis, both in nature, in enzymes, as well as in like industry, in artificial catalysis. And one of the reasons is that enzymes can exist in, metals can exist in different charge states. So there could be a switch from iron 2+ to iron 3+, so it can accept or donate electrons very easily. And it can also form some unique bonds with the substrate. So that's why metals are very popular in enzymes as well as in some industrial catalysts. Iron you can also see in some enzymes and proteins in such a form where iron is bound to sulfur, to such a iron sulfur clusters. So the simplest one is like single atom of iron somehow bound in, caged in some cysteines, or you can find such a 2 iron or 4 iron clusters. So these 4 iron clusters looks like some cube, and you can find it either in oxidoreductases, where it can again like exist in a 2+ state or 3+ state, or some other enzymes like aconitase in Krebs cycle uses this kind of iron sulfur cube. And I also added here a glutathione. So glutathione is like main antioxidant in the cell, and it's a simple peptide with 3 amino acids. There is quite strange like a peptide bond that this this first should be glutamic acid, not glycine but glutamic acid, so it's gamma glutamyl cysteine or glycine, sorry for that. So the first one should be glue, and this glutamic acid is not bound via this first carbon or first carboxyl group but via the carboxyl group on the side chain. So this is like difference from normal peptides. And important is that glutathione, it can exist in oxidized and reduced state. So normally it is a reduced state, like a single molecule of glutathione, and it can be oxidized on the sulfur, and 2 molecules of glutathione can join together to form like a disulfide bound dimer, which is the oxidized state. So mostly glutathione is like participating in some either enzymatic or non-enzymatic reactions where it is oxidized. So there are reactions in which some reactive oxygen species are converted from something which is less dangerous for the cell, and glutathione is oxidized, and there are enzymes which can reduce glutathione back to its reduced form. So for example, it is glutathione dehydrogenase, glutathione reductase, which uses NADPH to do this job. Now we switched to transferases, and quite important coenzyme of transferases is ATP. So it's not only the source of energy, but it is also a source of phosphate. So enzymes called kinases use ATP as the source of phosphate to phosphorylate something, and I will talk about many different kinases. You can find some activated monosaccharides, activated phospholipid heads, or some other molecules, like for example UDP glucose, UDP galactose, and some other molecules where there is some nucleotide diphosphate, and on the second phosphate there is something bound like a monosaccharide or a phospholipid head or some other moiety, and there are some transferases which can use this substrate, so it's possible to see these molecules as coenzymes. Quite interesting coenzyme is PAPS, phosphoadenosine phosphosulfate, which is, it looks like a little bit like ADP, but it must be, you should mention that here there is no phosphate, but there is a sulfur. So it's like extra phosphorylated, here extra phosphorylated ADP, but if you look closer, this is not phosphate, but this is sulfur. So it's a donor of sulfate anion. And what is sulfate used for? So there are some reactions in the body which need sulfation, and one of them is synthesis of different sulfated carbohydrates. So for example, heparines are example of such sulfated, heavily sulfated carbohydrates. So quite interesting molecule is S-adenosyl methionine, and this is a donor of methyl group, so it's methylating agent in body. There is another methylating agent which are folates, and the metabolism of S-adenosyl methionine and metabolism of folates is somehow like linked together. There is some overlap. And the same time, S-adenosyl methionine is like intermediate of methionine metabolism. So any intake of methionine in body, if this methionine is not used to synthesize proteins and it's metabolized like energetically, it goes first to, it condensates with ATP to form this S-adenosyl methionine. This S-adenosyl methionine is used as a source of phosphate, a source of methyl group in some methylating reactions. The product is like this molecule without this methyl group. So if you imagine like this methyl group is absent and there is no plus charge on sulfur. So this is S-adenosyl homocysteine, and this is hydrolyzed to homocysteine. So homocysteine is the same like cysteine, but it has extra one carbon atom here. And this is like a linkage between like methylation reactions in human body and this the metabolism of methionine or metabolism of sulfur amino acids. Here you can imagine like that this is quite unstable, like this arrangement. So there is sulfur with 3 bonds and plus charge. Yeah, indeed this molecule is quite unstable. So if you want to, for example, run some reaction with S-adenosyl methionine as a donor of methyl group, it's not possible to like go to some website and to buy S-adenosyl methionine because it's unstable. So instead you have to prepare some reaction which forms this S-adenosyl methionine like in situ, for example from methionine and ATP. This is the most terrifying structure in all these slides, which is vitamin B12. And it's quite interesting vitamin which contains cobalt in the center of this ring or this system, which looks a little bit like porphyrin in heme, but there is just one direct linkage. There is not no carbon atom like here or here. So it's slightly different from heme. And it's very complicated structure. It can contain here like coordinationally covalently bound kyanide, hydroxyl group or methyl group, and it's quite interesting. This is like prosthetic group. It can, for example, participate in reactions like conversion of methyl malonate to succinate, which is a isomerization reaction. So methyl malonate is like malonic acid is like dicarboxylic acid with 3 carbon atoms. So there is carboxyl group, CH2 and carboxyl group. So methyl malonate is methylated on this CH2, so there is a carboxylic acid, then CH, then it's branched and there is methyl group, and on another branch there is carboxylic acid. So if it's methyl malonyl coenzyme A, there is coenzyme A bound on one of these carboxylic acid moiety. And this can be isomerized to succinate. So methyl malonate has 4 carbon atoms, succinate as well. So this is simple isomerization. But again, if you like imagine like how to do it in organic chemistry lab, this would be a nightmare. So that's why enzymes need such a complicated chemistry for doing this complicated rearrangement reaction. It can also participate, for example, in dehydroxylation of nucleotide to deoxynucleotides. So for biosynthesis of DNA, this is important. It's a very non-polar molecule, and therefore it cannot simply like circulate in bloodstream. It cannot be distributed by bloodstream as it is, but usually it's distributed bound on some protein carriers. So there are three main protein carriers like haptocorin, transcobalamin, and intrinsic factor. They are quite similar in structure, and they carry cobalamin, and they distribute it through the body, and they can also participate in some storage of cobalamin. It's a vitamin B12, so vitamin with the lowest daily intake, so it's in microgram quantities, like intake of cobalamin. So I already mentioned that folic acid is quite interesting because it can exist in not in just two forms but many different forms. So here are examples of these forms, and it can exist as a donor of different like single carbon moieties. So if you consume a folic acid from some food, it is in the body reduced to dihydrofolate, and then it's further reduced to tetrahydrofolate. And tetrahydrofolate can accept a formyl group, and it can this formyl tetrahydrofolate can act as a donor of formyl group. This formyl group can be somehow like condensed here and reduced to metanyl or methylene group, or finally it can be reduced to methyl tetrahydrofolate. So this formyl can be a donor of formyl. This metanyl and methylene could be a donor of like methylene, a CH2 moiety, and this methyl can be donor of methyl, so CH3. And it participates, for example, in like conversion of uracil to thymine. So this methylation of uracil to thymine, it needs this folates and some other interesting reactions. I would also mention that there is a drug which is called methotrexate. So methotrexate, as far as I remember, looks exactly the same like a folate, except that this hydroxyl group here is changed to amino group, and this NH group is methylated. And this is like a synthetic compound. Methotrexate was developed quite many decades ago, and it's a very important drug because it's used either to treat some cancers or to treat some some autoimmune diseases, because it can somehow block the usage of these folates in the body. And for example, in cancer cells need to synthesize a lot of DNA. For this they need thymine, and for this they need folates. So if you block the metabolism of folates by such a compound which looks like a folate but doesn't work like a folate, it's possible to treat cancer. And there is also it is also used in autoimmune diseases treatment, because it has also some effect like a immunosuppressive. Vitamin H or biotin is very interesting vitamin, which acts as a prosthetic group in some enzymes. So normally like biotin is here like COOH or COO minus if you prefer, and as a prosthetic group is covalently bound to either to enzyme or very often to some a special protein which is the part of enzyme and which is called biotin carrier protein, biotin carboxyl carrier protein, BCCP. And biotin works as a carrier of carboxylic acid, or of carbon dioxide, to be more exact. So here is like the original form, and here is the carboxylated form. So it can somehow catch and activate carbon dioxide for some carboxylation reactions, for example in fatty acid biosynthesis. Thiamine is a very interesting prosthetic group which can somehow decompose some alpha keto carboxylic acid, so 2-oxocarboxylic acid. So if you have some moiety here, for example methyl moiety, is then this is a pyruvate. So for example pyruvate can undergo some like cleavage here, decarboxylation here by enzymes which use thiamine diphosphate, and one of them is pyruvate decarboxylase. You can find pyruvate decarboxylase in yeast, because in ethanol fermentation pyruvate is decarboxylated to acetaldehyde, and acetaldehyde is reduced to ethanol. Or you can find it in pyruvate dehydrogenase or 2-oxoglutarate dehydrogenase complexes, where again like pyruvate or 2-oxoglutarate is decarboxylated, but then by some other reactions they are oxidized to corresponding carboxylic acid, which is then attached to coenzyme A. Very important coenzyme is coenzyme A, and this is how it looks like. What happens here happens on this SH group. So the SH group can be acylated, so mostly it carries some acyl, for example acetyl or some fatty acid or something more complicated, and this somehow carries this acetyl or acyl. So by one reaction it is acylated, by another reaction there is something going on the acyl, and finally this is like deacylated and it returns back to coenzyme A. Here is vitamin B6 is pantothenate, which is like this part, and this is pantoate, is just this part here. So it is, this pantothenate is composed of pantoate, pantoic acid, condensed with beta alanine. So if you imagine like alanine, but you make an isomer in which like this normally in alanine this amino group would be bound here, but here is bound here, and this is cysteamine. So both like beta alanine and cysteamine are decarboxylation products. So if you imagine one carboxyl group here, you would have aspartic acid, and if you have imagine one carboxyl group here, you would have cysteine. So they are decarboxylation products of cysteine or beta alanine. There is something which is called acyl carrier protein, which is a protein which in its structure has covalently bound some similar moiety similar to the coenzyme A. So coenzyme A, for example, participates in fatty acid degradation, and acyl carrier protein in contrast participates in fatty acid biosynthesis. Very interesting vitamin and prosthetic group is pyridoxal phosphate. So here is the structure of pyridoxal phosphate. Very often there is an aldehyde group here. Very often you can find in enzymes that there is some side chain of lysine, so NH2 group. And in the resting state, when the enzyme is not like working, this pyridoxal forms a Schiff base, something like here, with the lysine side chain. And then when the reaction starts, it has to like wake up. Then this Schiff base is broken. This pyridoxal phosphate is released, and it can undergo some reactions. There are many different reactions in which pyridoxal phosphate participates, but to make one example is like decarboxylation. So, for example, if you imagine having some amino acids such as alanine. So alanine would be like, if you imagine here is CH3 group and here is COO minus group, then like this is alanine bound as a Schiff base to pyridoxal phosphate. So here you got pyridoxal phosphate, then alanine comes from the solution and binds to this oxygen, or replaces this oxygen to form a Schiff base. So here is CH3 bond, CH3 group and COO minus group. Then there is some rearrangement of hydrogens, and then here is this Schiff base is broken. But in this case, because here we got CH3 group, here we got COO minus group, so this is broken to get oxygen on this double bond. So we obtain pyruvate. So the pyruvate is released, but of course, like, because it's prosthetic group, it must be somehow like regenerated back. So you end up with this pyridoxamine, and it has to be somehow regenerated to get back to the pyridoxal phosphate, and it happens by the reaction in which instead of pyruvate, it's like opposite reaction, but instead of pyruvate here, it is 2-oxoglutarate, which somehow replaces the pyruvate here. So you end up with glutamate. And this is the reaction catalyzed by alanine transaminase. So the reaction is alanine plus 2-oxoglutarate gives pyruvate plus glutamate. And of course we do not mention in this reaction pyridoxal because it's prosthetic group, so it's recycled within this catalytic cycle, so it's just a part of the catalyst. It can participate in many different reactions, not only in transamination, which I have just explained, but for example in decarboxylation of amino acids. So in 2 or 3 slides before I mentioned beta alanine as a decarboxylation product of aspartic acid, or cysteamine as decarboxylation product of cysteine. So here this is the way how these compounds are synthesized, that the reactions enzymes need pyridoxal phosphate as a prosthetic group. And it could also participate in racemization. So for example, in some antibiotics, some peptide antibiotics, you can find some D amino acids, and D amino acids are formed by enzymes called racemases. So they can convert L amino acid to D amino acid, and usually these bacteria which produce, or some other microorganisms which produce antibiotics. They insert these D amino acids into these antibiotics to somehow avoid to prevent hydrolysis of these antibiotics by like regular proteases or peptidases. And here this picture is quite funny. So this is like a pyridoxal phosphate here. This is a lysine side chain, and this is pyridoxal phosphate bound via Schiff base, and this is enzyme called phosphorylase, so enzyme which decomposes glycogen. And it's quite interesting that this enzyme uses pyridoxal phosphate as a prosthetic group, but nothing important happens on this aldehyde moiety, but instead, like, only important part of pyridoxal phosphate is the phosphate, and this is kind of example of some opportunism of nature, that the nature can somehow like use something which is available instead of building some complicated chemistry. And in last 2 slides or 3 slides I will mention some vitamins which are not related to enzymes, or not related directly to enzymes. They are not some typical like coenzymes and prosthetic groups, but simply they were discovered that if you don't eat enough of these vitamins you get sick, but later it turned out that it's not related directly to enzymology. So vitamin C can act as a reducing agent in this reaction, in which dopamine is converted to some norepinephrine, for example. So dopamine is oxidized, as well as ascorbate is oxidized by molecule oxygen, and ascorbate is oxidized to dehydroascorbate, which is like there is a single bond here, and there are double bonds here and no hydrogens here. So this is the oxidized form of ascorbic acid, of vitamin C. It can also act as a kind of very strange cofactor. So there is enzyme which hydroxylates collagen. So collagen is like a protein which is important for some connective tissues, and it's basically like long chain of some long polypeptide chain rich in proline. And to get the proper properties, it's necessary to hydroxylate this proline. So it's necessary to attach hydroxyl group on the proline side chain. And this reaction is catalyzed by an enzyme which uses oxygen as the oxidative agent, and there is iron 2+ ion which is in the active site, which is a prosthetic group, we would say, and it's kind of bare iron. It's not bound in some heme or some special like coordination system. It's simply just iron 2+, and iron 2+ is very sensitive to being oxidized to iron 3+, and for this purpose this enzyme contains this vitamin C molecule, which is bound like basically in the active site or in a vicinity of the active site, and its role is to keep this iron 2+ to be 2+ and not to be oxidized to 3+. So it's kind of helping cofactor, and it's classified as a cofactor, so it's neither prosthetic group nor coenzyme. It's some special type of cofactor. And of course, ascorbic acid is important antioxidant. And another like vitamin which is not neither coenzyme nor prosthetic group, but it's related to enzymes, is vitamin K, which is responsible for carboxylation of glutamic acid side chains in some blood coagulation factors. So there are in blood coagulation, it very often happens that some protease, cleaves specifically another protease, and by this protease gets activated, and then it cleaves another protease and another protease, and this is kind of cascade in which the signal is like boosted. So it starts with some one activation molecule, and it can easily, like this chain reaction can strengthen this signal. And for this hydrolysis, it's necessary that some glutamic acids in active sites of these enzymes must be carboxylated. So there is like, if you imagine the structure of glutamic acid, so on the side chain you got like carboxyl group, and if you go to this methyl group just next to the carboxyl group, methylene group, then on this methylene group it's necessary to add another carboxyl moiety. So you got like some two carboxyl moiety on one carbon, and there are some derivatives called warfarins, which are used as drugs for treatment of thrombosis, some excess of blood coagulation, and they are again like examples of antivitamins. So they are structurally similar to vitamin K, but they are not functional. And now these three vitamins are completely like unrelated, or never say never, but I would say they are quite unrelated to enzymology. So vitamin A is of course important like in biochemistry of vision. So in eye we have a receptor of light called rhodopsin, which contains covalently attached retinal, and retinal, when in a photochemical reaction, undergoes isomerization, and this isomerization sends the signal to the cell, and this signal is then transduced to brain. And retinal or retinol is also quite important signaling molecule, so it's kind of a separated role. So for example, in developmental biology, in tissue development, there could be some signaling by retinol. Tocopherols, vitamins A, are kind of antioxidants, and calciferol, vitamin B, is like a vitamin with steroid structure, steroid-like structure, I would say, and it's important for metabolism of calcium. So in some regulation of storage and usage of calcium in body. So that's all for vitamins, prosthetic groups, coenzymes and cofactors, and I look forward to see you on Thursday, so we can see each other, we can look at enzyme terminology, and we can then have a look also at the homework. So see you, bye bye, thank you for your attention.